Hello students, my name is Dr. Rituparna Saha. I am an assistant professor from the Department of Life Sciences, Christu Jayanti College, Bangalore. And today I am going to be discussing regulation of oxidative phosphorylation. Now, in oxidative phosphorylation, one thing you already know that electron transfer takes place in between four complexes. Coming to the fourth and the last complex is that there the electron transfer is ultimately happens where the electron acceptor or the terminal electron acceptor is molecular oxygen. Oxidative phosphorylation is regulated by two processes. Number one is when the hypoxic condition. Now under hypoxic condition, what happens is there is an imbalance that happens between electron transfer and how the electron acceptor works in the terminal complex, that is oxygen. So as a result of which, because there is no electron to transfer anymore, the oxygen ultimately starts producing reactive oxygen species or ROS. So under hypoxic condition, in order to prevent the formation of ROS, the cell has devised two very essential regulatory systems. Number one is the production of a factor which is known as hypoxia inducible factor 1. This is a transcription factor. This transcription factor is produced only under hypoxic condition in order to prevent ROS production by two different ways. Number one is this transcription factor HIF1 goes and synthesizes the enzyme pyruvate dehydrogenase kinase. This enzyme is actually utilized in order to phosphorylate the active site of pyruvate dehydrogenase enzyme. Now this enzyme pyruvate dehydrogenase is responsible for the conversion of pyruvate to acetyl-CoA, and the acetyl-CoA ultimately enters the citric acid cycle through which the electron transfers ultimately start. So by stopping the pyruvate dehydrogenase enzyme, no more pyruvate is converted to acetyl-CoA and therefore citric acid cycle stops. And as no electron transfer occurs, so therefore in oxidative phosphorylation, ROS generation is prevented. The second method by which this hypoxic inducible factor 1 works is by increasing the synthesis and production of an enzyme, that is protease. Now protease goes in and degrades COX-4-1 subunit that is present within the complex 4. Now this COX-4-1 subunit aren't able to actually work under hypoxic condition. So the protease enzyme goes in and degrades its entire subunit. After which it starts the synthesis of an alternative subunit known as COX-4-2 subunit. This COX-4-2 subunit are actually adapted in order to perform under hypoxic condition. So this COX-4-2 subunit ultimately goes in and replaces the COX-4-1 subunit so that whatever little electron transfer can occur can actually takes place without ultimately forming any form of ROS. So this is how under hypoxic condition the hypoxia inducible factor 1 prevents the formation of ROS. The second type of regulation that takes place in case of oxidative phosphorylation is through the concentration of ATP and ADP. The relative concentration of ATP and ADP controls the rates of citric acid cycle, pyruvate oxidation as well as glycolysis. So whenever ATP consumption increases, the rate of electron transfer and oxidative phosphorylation also increases. Simultaneously, the rate of pyruvate oxidation via the citric acid cycle also increases, increasing the flow of electrons into the respiratory chain. So, when the conversion of ADP to ATP lowers the ADP concentration, acceptor control slows down electron transfer and thus oxidative phosphorylation also comes to a halt. Now glycolysis and the citric acid cycle are also slowed because ATP is an allosteric inhibitor of the glycolytic enzyme phosphofructokinase 1, and also of pyruvate dehydrogenase. Now PFK1 is also inhibited by citrate, which is the first intermediate of the TCA cycle or the citric acid cycle. So what happens is when the cycle is idling, citrate keeps on getting accumulated inside the mitochondria, which is then transported to the cytosol, after which both citrate and ATP cumulatively together goes in and blocks PFK1 and ultimately stops glycolysis from happening. Thereby, when glycolysis stops, inadvertently citric acid cycle also stops, ultimately stopping oxidative phosphorylation. Thank you.